Hello, everyone, and welcome to episode 3 of MRS TV. My name is Andrea Godfrey, and I am here to tell you everything you need to know about this year's 2020 virtual MRS Spring and Fall meeting. Stay tuned. As we enter the third day of live events, make sure to visit the Hub for networking opportunities, our virtual photo booth, and a number of fun interactive activities and more. We have a full day of fall meeting tutorials combined with the continuation of spring meeting live symposia. Today also features the first spring meeting poster sessions and a special symposium on materials approaches for tackling COVID-19 at 12:30 p.m. Eastern. Fall meeting tutorial sessions today include the following, and make sure to check the live event schedule for times and links. And the following spring meeting symposia hold live sessions today. Again, check the event schedule for times and links. In addition to those spring meeting symposia, the following fall meeting symposia have sessions today as well. Now let's dive right into our exclusive interview with another one of our fall spring meeting chairs, Sabrina Sartori. Sabrina, thank you so much for joining us. There is a lot of work involved in being an MRS meeting chair. Why did you volunteer for the role?
First of all, to be able to work with colleagues from different backgrounds and expertise, as is possible with a society like the Materials Research Society. Why I volunteer is because I think by being one of the chair of the meeting, you have somehow the possibility to stimulate the conversations around specific directions, and therefore have quite a substantial impact on the material science community. That is why I wanted to have an impact on how the fall meeting could be shaped towards hot topics and maybe topics which have been less discussed but very current and of large interest.
What are some of the lessons learned from recreating scientific meetings now in the virtual space? And do you think that this experience will have a long-term effect on these types of meetings once we are able to actually get back to the in-person meeting experience?
Yeah, the first lesson I would say, I learned that it's not easy to organize a virtual event which is engaging. The reason why we go to a conference is typically because we can meet colleagues, or we can know that scientists that publish a specific paper that we found so interesting, and then at the meetings we can meet each other. In virtual meetings, this is a little bit more difficult. However, what we tried with our fall and combined spring and fall meeting 2020, we tried to give these special feelings of connecting and meeting. And this is a little bit different than in-person meetings. What we tried, how we try to recreate, is, for instance, offering spaces for one-to-one meetings after maybe a talk. And then this talk maybe is of interest for somebody and not for the general audience, and then people can have a meeting decide to have a meeting one-to-one and continue the conversation. And this way, we think this can engage. This can be an engaging meeting as an in-person meeting. We also tried working with the length of the talks. We made talks shorter than the usual talks at in-person conferences, and give more space for Q and answers, question and answers, and discussions. So we believe that with these actions we can engage the community as much as possible. And the second question that you asked, if do you think this will impact the future way we will do conferences in the future? It may. I think there is no way to substitute in-person meeting. However, we could continue and consider to propose at least one part of a conference as a virtual event, maybe with one or two symposium that could be, for instance, beneficial for scientists that may not be able to join in person.
Sabrina, thank you so much for your time today.
Thank you very much. Have a nice conference.
Let's continue our tour of universities and organizations in the world that are changing the world with their research.
The main mission of the MSK Cornell Center for Translation of Cancer Nanomedicines is to evolve a suite of ultra-small core shell silicon nanoparticles for a range of tasks, both diagnostic and therapeutic. We do this by the precision tailoring of the particle surface and controlling its size to be able to improve the biological properties of the platform and improve the therapeutic responsiveness in vivo. We aim to translate these particle probes for these different applications. This would go to the clinic and then move along a commercialization track. So with my partners at Cornell University, Uli Wiesner, and at Elucid Oncology, Gino Germano, we are advancing along several paths to move these platforms into patient care in the long run.
What we did for the first time is to engineer particles that can both target the tumor, can deliver specific cargo to the tumor, and can signal to the outside where they are, all within a single particle. Special thing about these Cornell dots is that they are really what we call ultra small. They're below in size the cutoff for renal clearance, basically a molecular size scale. This gave birth to this paradigm of target or clear. We either target the tumor or the particles get out, therefore reducing the off-target toxicity. This is a really, really important point, and we think that makes the technology really special. After having gone through multiple generations of particles, we now synthesize them in water. We do the synthesis in a style which we call one-pot. We basically, over the years, learned how to add the different molecules that we want to combine on the particle in order to generate the final particle.
So Memorial Sloan Kettering has worked with Cornell for the past 14 years to really build this platform for a variety of applications. We are really the translational arm and the early clinical arm. What we typically do is we receive particles from Cornell, and a lot of particle batches have been made at Cornell, and we screen the particles, select the lead candidate, and advance it for animal work. So we have moved to prepare these particles in a manner that can be injected into patients to validate what we've seen in our small animal models.
The novel technology behind CDOTs, it fulfills an unmet need. Right now, there is no technology that's available to the surgeon to be able to see an optical signal that's linked to cancer cells. We have been doing a first-in-human clinical trial using CDOTs in head and neck melanomas for the past few years. So far, the results are pretty encouraging. You get a very, very strong signal intraoperatively, just as the theory behind the platform is.
By tailoring all of these properties, we would then be able to move this into clinical trials, and then move it along a commercialization track, which is where we are now.
At this point in time, Elucida is focused primarily on developing the platform as a delivery mechanism for therapeutics. We have a lead compound that's a folate receptor alpha targeted CDOT drug conjugate. And we've seen some promising evidence so far in preclinical studies that we may have some pretty significant advantages over the standard of care. So we're very excited about that.
I was a PhD student in Professor Uli Wisner's group at Cornell University, the Department of Material Science. Since 2009, I've been working on the platform at John Elucida because we really think this platform have a lot of potential to really make a big dent in the field of drug development and cancer treatment, and also be promising to potentially help a lot of cancer patients and improve their life.
The center has evolved a number of probes for various applications, and I see the future as linking this technology and partnering with other technologies.
I think the CDOT platform represents the next frontier in targeted drug delivery, in cancer therapy.
I think we've come a long way. There are so many exciting new directions that we're going to be busy for the next decade for sure.
Now let's head to California to get a closer look at all the work the COP Lab at the University of California, Irvine is doing to advance the field.
The main mission of my laboratory is to understand how macromolecules can encode information that can be used to self-assemble nanomaterials with custom properties. We're also interested in using these self-assembly rules to make novel photonic and electronic nanomaterials. My research laboratory is unique because we are an interdisciplinary group of scientists with students from chemistry, physics, material science, and chemical engineering backgrounds. And while we primarily do experiments to study materials, we also use tools from data mining and machine learning. This is because the soft materials we create are very difficult to model from first principles. So we need to have alternate ways to learn the physics and chemistry that describe how self-assembly happens in macromolecular systems. Most of my research right now focuses on how DNA can be used as a programmable molecular building block for nanomaterials. Specifically, we're using DNA's nucleobase specific interactions with certain metals to create the tiniest of nanoparticles, which are called nanoclusters, and also to create tiny atomic wires of metal. In addition to this work on DNA, we're also interested in using other macromolecules like block copolymers and peptides, all for the goal of creating self-assembling photonic or electronic nanomaterials. One of our current projects that I'm really excited about right now is to develop novel fluorophores which emit light in the near-infrared spectrum. The reason why this is important is because biological tissues are much more transparent to near-infrared light than they are to visible light that we can see with our eyes. So by creating tiny molecules or nanoparticles that can emit near-infrared light, we could use these as biolabels in order to image deep inside tissues or even whole organisms. By building off the approaches that we've developed for designing visibly fluorescent silver clusters, we're now turning towards the near-infrared spectral region, trying to design the largest silver clusters stabilized by DNA that have ever been made, because these will emit within the near-infrared tissue transparency windows.
Since we are using machine learning to try to discover new DNA-stabilized silver nanoclusters that have emission wavelengths in the near-infrared, we want to have large data sets so that we can train the machine learning algorithm. And that's why we're using high throughput experiments. And we are using the robot liquid handler so that we can produce many different samples in short periods of time. We are synthesizing 384 different clusters in about 20 minutes. We are also using the robot so that this way we can reduce the amount of time that we need to synthesize them, and also reduce the variability between samples due to human error.
The DNA we use to stabilize our silver nanoclusters is 10 to 30 bases long. This means that there's a huge number of possible DNA sequences that we can use to stabilize our clusters. We use machine learning to try and predict the properties of these clusters so that we can create targeted synthesis and try and synthesize clusters, allowing us to save time and money and not having to sample the entire sequence space for the clusters we're looking for.
The DNA sequence will determine the size and shape of the nanocluster through the different interactions between the 4 different nucleobases with the silver. And the total number of reduced silver atoms will determine the emission color of the nanoclusters.
My group and a lot of other people around are very interested in characterizing new hormones, endocrine factors that circulate in the blood. And so we got really excited by Stacy's finding these dyes that they've developed, specifically because we think that it could give us the ability to tell us where these molecules are going in the blood and the body and where they're acting upon.
In the long term, my vision for my research laboratory is to be able to understand, in general, how macromolecules encode chemical information that can then be used to drive organization on the nanoscale. I envision that we could be able to create novel nanomaterials that we can't access right now with the solid-state materials paradigm, to create things that are dynamic, that can interface directly with the body, and that can create and manipulate light in ways that we can't achieve right now.
Thanks again for tuning in, and we hope you enjoy the rest of the meeting today. We will see you tomorrow.
Thank you.